So good morning, everyone. Thank you for joining the Oregon Bioscience Association at today's BioBusiness and Good Beans Zoom Coffee Talk. Hope you've all grabbed your cup of joe and are ready to go. This morning, we'll be discussing research and therapeutic innovation in response to COVID-19. In this time of COVID-19, info is rapidly changing and so is the speed of business. We want to help you keep updated on the topics of the day and how you and your colleagues can benefit. Today, Oregon Bio, in our 30-minute fast track touch question, we're going to spend some time with Drs. Fox and Barr-Gillespie. Before we jump into the session though, I want to take a moment to acknowledge and thank our sponsors of today's Coffee Talk, Commissioning Agents Inc., Skanska, Summit Development Group, and VWR Aventur Sciences. So here's how our session is going to go this morning. We'll first hear from both Drs. Fox and Barr-Gillespie, and then we'll open it up for Q&A. We ask that you submit your questions via the Q&A button or the chat screen. You can also hit the button to raise your hand. And if we're not able to get to all the questions in our time together, please submit them via email to news@oregonbio.org after the fact, and any remaining questions in the Q&A or the chat, we'll make sure to get answers to those and post those and send all that information back out to our attendees. Also, our program is being recorded and will be online at oregonbio.org/events for the next couple of weeks. So now it's my pleasure to introduce a couple world-renowned experts. Bernie Fox wears many hats. He's a translational cancer immunotherapist at the Earl A. Childs Research Institute at Providence Cancer Center, a biotech entrepreneur, is the CEO and founder of UbiVac, an adjunct faculty member pursuing molecular microbiology and immunology at OHSU's Knight Cancer Institute. He spent more than 30 years studying how to use a patient's immune system to fight cancer. Dr. Fox has led both preclinical and clinical studies of cancer immunotherapy. He is the current chair of the World Immunotherapy Council, a consortium of 22 national and international cancer immunotherapy organizations, and a past president of the Society for Immunotherapy of Cancer. Following Dr. Fox, we'll hear from Peter Barr-Gillespie, Ph.D., who is the Executive Vice President and Chief Research Officer at OHSU. In addition, he's a professor with the Oregon Hearing Research Center and an affiliated scientist with the Volum Institute. As an NIH-funded investigator, Dr. Barr-Gillespie's active research focus is understanding the molecular mechanisms that enable our sense of hearing. Specifically, the Barr-Gillespie lab endeavors to determine how sensory cells in the inner ear, called hair cells, allow humans to perceive sound arising from the outside world. Dr. Barr-Gillespie is also the scientific director of the Hearing Restoration Project. With that, I'll turn it over to Dr. Fox for this morning's conversation on the science behind COVID-19.
And I'm going to hopefully get my slides up. Can you see my slides?
We sure can. Thank you.
Okay, so I thought what I would do is take you through a focus in the time that I have on really the vaccine development and the way we've been seeing it from I would say 47th Street over here in Glisan. As part of my conflict of interest management with my institution, I have to disclose that I have actually relationships with different companies. I don't know why that's not going to the next slide. There it goes. So the only ones that are really important here are really the UbiVac. So I'm a co-founder and CEO, uncompensated CEO of UbiVac. And I have a collaboration with OncoSec, which we're collaborating with on this vaccine study. So this is from the 9th of April. This is Nature Review's summary. But as of the 8th, there were 115 vaccine candidates, 73 in exploratory preclinical studies. But there were only 5 as of April 20th. The World Health Organization identified there were only 5 that were actually in clinical development. Those 5 are shown here. So the first one that entered in the US was the Moderna study. It's a phase 1 clinical trial of the mRNA for the S protein, which is a component or the spike what you see, which is sticking out of the virus pictures, that spike is what binds to a receptor on human cells and allows the virus to get into the cell, or at least starts that process. So that trial is already enrolled about 45 patients, and that's the phase 1 clinical trial. Moderna is expecting to launch their phase 2 clinical trial from press releases either late spring or early summer. The study that's the furthest along is one from CanSino Biologics, which is a Chinese biotech associated with their governmental agencies, but it's an adeno-5 vector, adeno-5 vector. They've got now several, I think almost 200 people that have been injected with that. That was entering phase 2 clinical trials this week. I don't know if that actually happened, but I bet it did. Inovio has got another phase 1 trial. This is, again, the S protein. Again, that's a spike protein that allows the virus to get in. That's entered phase 1 clinical trials. There are 2 other clinical trials that have started in China. Both of them have used antigen-presenting cells, which are the cells that help orchestrate an immune response. And there's less information known about the specific mini-genes that they're using. So this is where the status, there are actually been press reports about other companies that are going to be and academic centers entering the vaccine space. But as of a couple of days ago, the World Health Organization had yet to announce that those had actually started. So I'm a cancer immunologist. I've been working in cancer vaccines for many decades. And we think about things differently than viral immunologists do, viral vaccine people, because cancer vaccines tend to be weaker antigens or things you've got to work harder to get an immune response against, and they're not as foreign looking as a virus is. So I'm not sure if this is better or worse, but we certainly look at them differently. And it's not just about vaccines that we think about things differently, but I think it's about how you treat patients with a viral infection. So in fact, my colleagues and I from actually around the world, including Europe and China, put out an editorial a couple weeks ago already, which just talk about some of our insights that we have from the immuno-oncology space to that. And this is an open access article. So we took those ideas and we wanted to put together a vaccine for the virus. And this started actually March 10th. And what we did is we built on work from the NIH. So this is a virus spike. This is a cryo-EM image of what that spike looks like. This little green thing at the top is what binds to the receptor on the human cells. So essentially, we got access to this spike through a collaboration with OncoSec. And then my colleagues, Dr. Ram Widener is actually leading the clinical trial. He's the principal investigator. He put the trial together. My colleague, Dr. Hongming Hu, who's the genius molecular biologist, put together the plasmids and things in short order. And then we worked with OncoSec, we didn't want to just give the virus alone, or the viral vector, viral plasmid alone. We wanted to combine it with an adjuvant. So they had a clinical trial with IL-12. They also have technology which, once you just inject the plasmid into, or the piece of DNA that codes for the virus spike into somebody, that alone is not going to be enough to get a good response. You want to be able to boost expression. So by you're adding electric shock to the spot where you put the vaccine in, you can substantially increase the amount of expression of that protein, which means you should be able to get a stronger immune response. So we're doing that, plus we're adding IL-12, which really boosts the destructive immune response. It's one of the molecules, the immunological hormones that can actually direct an immune response towards a real destructive immune response, and also boost antibody responses, which potentially can help neutralize the virus and eliminate it from the body. So we had the genetic information from the NIH, we had the clinical trial expertise, we had other technology that we could use to boost expression. But the problem was, to get a plasmid made through a CRO would have taken about 5 months at a minimum to get it made in GMP status. So UbiVac, which has been around for 15 years and has had a lot of GMP experience, particularly over the last year and a half, actually took on the task of making the plasmid GMP using the technologies that we have. So this is essentially what we're using again. I don't know if you can see the whole spike or if my picture is hiding it, but that spike is what we're immunizing people with. Essentially, it's this part of the DNA of the plasmid. This is the design of the clinical trial and what was submitted to the FDA on March 30th. It is a collaboration. Again, you can see Dr. Leidner is the principal investigator, but we have infectious disease doctors. We have other medical oncologists that are used to phase 1 clinical trials and first-in-human studies. We have scientists, and we have a scientific collaborator, Dr. Twitty from Oncosec. It is really, there are 2 major arms. Patients are either going to get vaccinated, actually these are normal volunteers, are being vaccinated with either the virus protein by itself, which is similar to what the individuals received in the study, the Moderna study up in Seattle, or they're getting that same viral spike along with the plasmid for IL-12. This is a cohort of patients which are the normal people, which are 18 to 50. This is normally where vaccines go, into that age group. They don't typically go into the older groups, but we had a great interest, particularly because the immune responses to coronaviruses tend to be weak at the beginning, and people as they get older tend to have weaker immune responses. So it won't help if you develop a vaccine that works in young people and then try to give it to old people. So we've pushed for the FDA to be able to enter into patients that are above the age of 50 with no cap on the upper age. And we'll see what happens. We have a few more days before we get final results from the FDA, but we hear informally that the FDA does not have a perspective on limiting the vaccine at this point. So that's what we're going to be doing. Patients will get 2 vaccinations. They will get one at day 1, and at day 30, there will be blood draws to assess the immune response. So this is an outline of how fast this went. Usually it's taken us 6 months to a year to get an IND put together. This went together in 20 days. We came up with the idea on a phone call. We went through the whole process of licensing the plasmid with our colleagues at Oncosec, getting IBC and IACUC approval, started animal studies, and have safety data already on this, as well as the safety data that we had from the NIH. UbiVac has gone through making the master cell banks and manufacturing GMP plasmid, manufacturing that, et cetera. So if you think about vaccines, you're hearing all these stories about it's going to be 12 to 18 months, 16 to 18 months. This I'll just steal from the analyst couch from the Nature review about the time it might take. They think there may be similar protocols that will be available for emergency use in early 2021. I think that's not true. I think what you're going to see is you'll see emergency use authorization for vaccines that have safety profiles, and you'll see them for first responders and healthcare workers likely in the fall of this year. So I think you're going to see vaccines moving very quickly if we can get the right safety data and evidence of immune responses, which I think we will get. That what I've talked to you about is certainly not the work of just UbiVac or Providence. We have colleagues at Oncosec, but I have to again just point out that Ram Leidner, my clinical colleague, is really leading the charge. I'm inspired by his interest in pushing what we know immunologically into patients. I don't know what I just did there, excuse me. But putting it forward, and also my colleague, Dr. Hu really had the perspective of the molecular biologist. But I'm sorry, hopefully this is going to come up now. There, I won't touch my slides again. But there were other people, Sean Jensen, another OHSU grad who went on, has been leading the charge on the preclinical studies in the animal to get the safety data that we needed. Chris Twitty, who's now the chief scientific officer at Oncosec, who's also a PhD graduate student or grad from OHSU, who did his PhD in my lab and is now the chief scientific officer there. My colleagues here in molecular that are doing all the sequencing for us and all the sterility testing for us, and the regulatory group. But I can't say enough about the people at UbiVac, and Tracy Hilton's name is left off there, but these people have been working kind of around the clock to push this. But I'm a cancer immunologist again, I say this, and you look at it, we went into this without much experience, but we've had the training of how to do these things. We've had the experience of first-in-human clinical studies. We had the resources in the facility, and I had donors. There were philanthropists who I knew would support this. And in fact, a few days after we announced this, Nancy LaMotta wrote a check for $2 million. And if it wasn't for that, we wouldn't be able to do these sorts of things. But it's also the vision of Walter Erba, who essentially designed this institute to be first-in-human sort of studies 26 years ago, and has supported the infrastructure that allowed us to do this. Because you can't just do this in a regular lab, you've got to have other resources and support. I think my 10 minutes is up, so I'll stop. And I'm going to go back to stopping sharing. I went through that all very fast.
That's great. Thank you, Bernie. Peter, I'm going to turn it over to you, and then just hold. We've got some questions in the session here, but we'll hold all those to the end.
Hey, thank you. That was fascinating, Bernie. Thanks for presenting that. That is really exciting. I'm going to talk about OHSU's response to COVID-19. And as you can imagine, as the state's academic health center, we've been extensively involved in responses. One of the first things that we did was to set up a test for COVID-19, SARS-CoV-2, and we actually had to set up our clinical lab in order to do that, and we managed to pull off both starting the testing and setting up the lab in a couple of weeks. But the lab is great not just for testing patients and employees and others, but also we're using the lab to develop new assays, including faster and cheaper assays for testing for the virus for active infections. And then also serology tests to test for antibody levels to see who has been infected in the past. We expect to roll those out relatively soon. So, you know, at the same time we were setting up the clinical lab, we actually had to shut down research at OHSU. 1,200 labs had to close, and we had to do that incredibly quickly. As you know, that's happened around the country and in fact around the world. We did that because we needed to protect our research staff, conserve PPE and other reagents for the hospital, and reduce the logistics load on the university, which again supported the hospital. And as you know, the actions that we took, but then of course the whole state took, have tremendously blunted the response, the infection rate of COVID-19 in Oregon, which has been great. So even though we shut down over 1,000 labs, we also recognized that we couldn't shut down those folks who are ready and raring to go working on COVID-19 projects. So we took advantage of the expertise at OHSU in infectious disease, immunology, vaccine development, therapeutics, clinical trials, structural biology, and we got a task force put together that would review potential COVID-19 research, and not only review the research just for appropriateness and whether we should allow the people to come in to work in the lab, but also to try and connect people together who are working on different parts of responses that might collaborate. So I was just looking at the registry of all the projects that we have right now, and it's up to 170 different projects with probably more like like 120 different investigators or more, and that's just in a matter of a few weeks. So we've been focusing on some of the areas that Bernie has talked about, like vaccine development. I think the vaccine development work that's going on at OHSU is more of the slow and steady. The work that Bernie is working on is fantastic in that it's so much closer to going into clinical trials. I think anything at OHSU is going to take some time, but we have a variety of labs that are focused on that. As you know, we have the Vaccine and Gene Therapy Institute, the VGTI, which is designed for this. They have world-renowned experts in vaccine development and understanding viruses, et cetera, and they are working hard on a variety of different viral vaccine approaches. As our scientists at the Oregon National Primate Research Center, the Primate Center right next door to the VGTI, both of them are out at the West Campus in Hillsborough. And you know, as I say, that there are multiple vaccine approaches that will take some time to put together. We also have the primate resources at the Primate Center, not just the folks working on vaccines. We don't have any infected monkeys at the moment right now, although, as you know, the rhesus macaques can be infected by SARS-CoV-2 and develop a mild COVID-like disease. We expect to use those monkeys only for the most important experiments with COVID-19, but certainly development and testing of vaccines is going to be one of the most important uses for the monkeys. Also along those lines, we have another fantastic resource. Bernie referred to the cryo-EM structure of the spike protein on the virus. And we have a national cryo-EM facility at OHSU and cryo-EM experts here. And there are a variety of groups that are working on structures, not just of the spike protein, as you saw, that's already been determined, but other structures that are going to be important for understanding how SARS-CoV-2 infects cells and hijacks them, but also in figuring out strategies for vaccine development. So in addition, there are other things we're doing. There are lots of faculty who are developing or testing new therapeutics for COVID-19. And we need therapeutics because we need to manage the disease and the time between now and when we have effective vaccines that are widely distributed. And so there's a lot of different clinical trials or new therapeutic development going on. There's even a clinical trial testing the notorious hydroxychloroquine going on at OHSU, but other ones as well. There's one to highlight is that investigators at the Knight Cancer Institute realized that one of the cancer cell markers that they've been targeting, a protein called TMPRSS2, is in the exact same pathway that the virus uses to get inside cells. And they're looking to see whether they can repurpose some of the drugs they use for prostate cancer in this case, to see whether those drugs might in fact reduce virus entry. We don't know, but we'll see. There are other faculty who are developing novel drugs to reduce the impact of the disease, using chemistry to modify pre-existing drugs, or screening for new drugs using different chemical screening methods. There are other creative ideas that our faculty are taking, like Albert Chi, who's a trauma surgeon. He's developed a 3D printed ventilator that can be made from just $10 of material, a 3D printer, and a few springs from a hardware store. And that plus an oxygen tank gives you a functional ventilator, which will be certainly important if we have a second wave of disease and we run into a ventilator shortage again. But even more important, I think, is that this 3D printed ventilator can be deployed in developing countries which can't afford the tens of $1,000s ventilators that we typically use. So also the OHSU PSU School of Public Health has been approaching the pandemic with their particular expertise in epidemiology and community research and social determinants of health. And in fact, I've asked the Dean of the School of Public Health, Dr. David Bangsberg, to lead a joint OHSU PSU COVID Council, which has been charged with developing big ideas on how to attack COVID-19 around testing and surveillance, around vaccines and therapeutics, and also around the social and economic consequences of the COVID-19 pandemic. So just to sum up, we've got amazing resources at OHSU and lots of expertise. And we're working closely with scientific collaborators around the world on different projects, but we're also working very closely with the state, especially the governor's office and the Oregon Health Authority, to make sure that our state's response is guided well. So thanks.
Thank you both. It's been great overview of what's going on in our region here. And I think the first question is, when do either of you sleep?
Yeah.
Yep.
Not as often as I'd like.
I try and sleep as well as I can because we're in this for the long haul. So,
Yeah, okay. Well, I'm going to get to the real questions here. any work at OHSU on blunting cytokine response syndrome or cytokine storm?
Yeah, I mean, there are a number of different approaches that people are taking. I couldn't tell you them off the top of my head. I know that in that list of 170 projects, there are a number that are focused on cytokine storm. I can follow up later with specifics, but yes is the answer.
If I can comment, if somebody's interested in that, they can go to SIT Cancer. It's just Society for Immunotherapy of Cancer. There's a preprint there that lists and goes through the IL-6 pathway, which is the main pathway, and a drug that's likely to be effective, as long as a list of a series of other drugs that are there. And that has been peer-reviewed and is about to be in press, but a preprint is available on the SIT Cancer website.
Great, thanks. I've seen some discussion about younger vaccinated volunteers being challenged with the coronavirus to help speed up the process for determining efficacy. Do you think that will be an approach taken in the US?
I'll let you take that first, Bernie.
Yeah, you know, that these vaccine challenge studies, you know, there's an ethical debate around them, and I'm not sure where it's going to all fall out. We are not planning to do that in our center. And we've got like second and third generation vaccine strategies that we're pushing on, but we're not planning on that kind of challenge.
I would imagine that is absolutely going to be the case at OHSU as well. And that's one of the reasons why we would use our primate center for testing some vaccines that haven't been tested in humans, just to get a preliminary look at the safety and effectiveness before going on.
Great. Once the vaccine is approved, what's the timeline for scale up and broader global distribution?
You know, it's we're hoping to launch the phase 1 by mid-May. We've got we're actually thinking more along the lines of, while we're preparing to go to a phase 2, if that's like blockbuster responses, but we're already looking at doing 2 other phase 1 studies, which would be different, and others using one that uses UbiVac's kind of platform technology, or the dribble technology, as well as other protein type, like altered small viral particle type strategies. So I don't know when that would be, but we're thinking you could certainly scale up to large phase 2 trials late summer, early fall.
Great. I want to be mindful of the time here. We have a few questions left in the chat. It's almost 10 o'clock. I want to just do a quick thank you to you both for your time today, and give a quick shout out for our sponsors for making today's program possible. Again, it's commissioning agents, Skanska, Summit Development Group, and BWR Aventor Sciences. And also, just for the folks that need to drop off at 10, let you know that we've got a special program planned next week. It's Cinco de Bio. It's going to be on May 5th. It's going to be a happy hour format, and it's going to be our BioBusiness Solutions partners and some fun networking opportunity in the afternoon there. So check out oregonbio.org/events page and get registered for that later today. It's 10 o'clock. I think our panelists agreed to hang around for a couple minutes to get through some of these questions. Are we still good for that for next few minutes? So anybody that can hang around, please do. We'll keep going on the questions for a little bit here. If you can't, feel free to check out our YouTube page or our events page later today for the video recording. You can catch the tail end of today's session. Let's see, there's been some discussion about needing more than a protective antibody response. Any thoughts of including a cellular response, NK cell?
Yes, no, absolutely. That's the IL-12 will actually drive a cytolytic or a killer T cell response, as well as boost the innate immune system, which includes the natural killer cells. And so it's becoming more and more clear that that's likely important in patients who have cleared virus but have got very, very low levels of antibody responses.
How were the antigenic epitopes for vaccine development selected, and how were the titers screened?
So the spike was one that was selected. That's one that overall we know that from the other SARS that an antibody against the receptor binding domain of the spike can neutralize. And so it was selected there. There's also correlation between antibody responses and T cell responses to the same epitopes. There has been, non peer reviewed yet, but in medRxiv, there's several papers from Chinese investigators that correlate T cell responses against the nuclear protein again, and correlating with neutralizing antibody titers. So the first wave of the vaccines I showed were the first 3 that are in large scale studies or early phase studies are all against the spike. That only includes that component. I think next generation vaccines that we're looking at will include other parts of the virus as well, and with potentially just T cell epitopes, so you don't have this antibody dependent enhancement, which may be a negative impact of a vaccine.
Okay, there's a question in here. What can we learn from the Ebola vaccine and fast track learning from that development to help expedite the current process? Isn't the virus/flu vaccine development standard development process? We aren't really inventing the wheel again, right?
So but I think the issue there is that the coronaviruses tend to get very weak responses, and they tend to fall off very quickly. The IL-12 has already been tested in the Ebola, so the strategy we're using is one of the strategies that has been used in Ebola to get boosting immune responses. But I think that in general, responses are weak. And so I think we have to think about this differently. And so again, cancer vaccines, immune responses have been weak. And for decades, we've tried lots of different molecules. We are doing the same thing now. We have IACUC and IBC approval to test multiple different strategies that we're using in patients. So we have, UbiVac is with a partner, we'll announce this next week, as a partner that we're partnering with to do a multi-center study in advanced triple negative breast. We're adding other agents in. We're looking at putting some of these same kinds of agents into our vaccine studies, but in a plasmid level instead of having to give IV administration of drug. I think that's kind of the innovative thinking that I think will allow us to potentially get much stronger immune responses, because the majority of what you've heard from the large pharma companies are going to be standard vaccine technologies. And if they fail to induce strong responses, they may not be as effective as we would like. And I would think that the technologies that also, shout out to OHSU, the work that's been going on there with CMV viral vectors and the work that they're doing is really amazing technology. And that's a very strong, potent vector system. And I imagine that Klaus and others over there are working on those very things.
Yeah, that's right. So the CMV platform, which has been developed for TB and HIV vaccine development, is being repurposed for SARS-CoV-2 as well. And I think, you know, as Bernie said, the response of vaccines to the coronaviruses and the vaccine responses have not been great. As a matter of fact, with SARS-CoV-1, you know, it was found that in some cases, the adjuvants, which are used to boost the immune response, actually led to an enhancement, an increased level of disease, which is not what you want with a vaccine. So there's a lot of caution around different approaches to vaccine development, and this will all shake out during the standard development of the vaccine. But the other thing is that, of course, there are many coronaviruses out there, including some that cause the common cold. And while we get some immunity to those, the immunity is not particularly strong. So the idea that we can get herd immunity by just getting everybody exposed to COVID-19 may or may not hold. I mean, this is why we need multiple approaches. You know, the strategies that Bernie is talking about for boosting the potency of vaccines, but also therapeutics, I think are going to be really important.
Absolutely.
And just to circle back to the question, you know, remdesivir, which has been in the news a lot in the last 24 hours, because it seems like maybe it is at least partially effective at reducing some of the COVID-19 symptoms, was developed against Ebola virus originally. And one of the strategies, of course, with vaccine development or therapeutic development or what have you, is to look very carefully at the response, the cellular response to the infection and target, potentially target, in the case of therapeutics, target pathways that are common in the cellular responses to multiple different viruses. So maybe there will be more drugs that can be repurposed, and the focus certainly will be on drugs that will potentially be targetable against multiple viruses.
Super, thank you. It looks like we're down to 2 questions and they're pretty similar here. So I'm just gonna, how far are we from a more universal vaccine platform that could use the non-mutating stalk of the spike protein instead of the constantly mutating green colored top protein we showed in the images of the ACE2 binding spike protein? Will the effort move in that direction soon?
So I think that the entire spike has been pretty constant from the 12,000 or so that have been sequenced. So I haven't seen that mutate a lot. So it has to maintain a certain configuration or the virus won't be able to bind to the ACE2. But I also agree with the issue about the stalk part of it, right? There's part of the stalk that we think that if antibodies targeting that may not prevent or neutralize the virus from binding, and may also support the negative effects of a potential vaccine, or what they call the ADE, or antibody dependent enhancement. So I think strategies that you're not going to see happen from the big pharma, right? But maybe you will. I don't know. We'll be just looking at the green part of the associated part, which is the critical element to get the virus into the cell. And so you may see that there's going to be strategies that are going to target just that. That's only going to be good, likely, for this coronavirus, because you could have other coronaviruses that could bind other parts of the ACE2 receptor, but at least it would be more effective against this one. Yeah.
Okay, sounds good. I think that pretty much closes us out here. Oh, let's see here. I got one last one that just popped in. What is your view on convalescent serum, either patient-derived or recombinant?
Go ahead, Bernie.
So we're actually we're part of 51 hospitals, right? So the original first U.S. case, it was in Everett, and so the Swedish group and that in Providence, as of yesterday, had treated 32 patients with convalescent sera. It's way too early. On another call we were on, the Newark group, I think, has got the most. They were at 37 patients treated as of Wednesday morning. So we know that patients are getting sera. The big thing is screening, developing banks of sera. There's not good assays to assess this. It's taking too long to get the viral neutralization assay, even done from NIAID. So the people are doing different tests. The FDA guidance calls for negative RNA and a positive antibody response. So those are the requirements for the FDA IND, as I understand it. We're running the Mayo Clinic protocol through our 51 hospitals for that, and looking for people who have got antibody titer. I will just say that we've screened now more than 9,000 Providence employees for antibody to that, and are finding a very low percentage of people that have actually picked up the virus, but have found people, and those are people that are being contacted to see about sero donors.
Right, I think that really highlights one of the challenges with using plasma. It's just the low incidence, certainly in Oregon, of COVID-19. So the amount of plasma available is going to be limiting for a long time.
All right, well, I think that really does close us out here. Thank you both so much for your time. This has been really informative, and I know our audience has enjoyed it immensely. So very much appreciate it. Thanks for all the good work that you both do. And also thanks one more time to our sponsors for making today possible. I'll list them again. It's Commissioning Agents, Skanska, Summit Development Group, and VWR Aventur Sciences. That's a wrap, everyone. Thanks so much.
Thank you.